my screen share here and fire this up. Now, are we seeing which, let's swap these, right?
Yes.
Perfect, okay. So first of all, I've been watching the chat and I'm seeing how many people are from so many different places. It's very cool. I mean, it's, you know, it's hard because of this virtual presentation. I like to be able to see people's faces, particularly if I'm about to confuse a bunch of people. But in this case, it's really cool to just see everyone chatting from all over the world, really, all over the country, all over the world. I saw Poland on there. Shout out to Poland. So I really want to thank Nicole and the MDA for giving me the opportunity to speak with you all today. This is, you know, I feel like anytime I hear these update talks, everyone says, oh, it's so exciting, and it's a bit of a rah-rah. And we become there's a risk of sounding enthusiastic each year and becoming the boy who cried success. But I really feel like there is no better time to give an update talk. It's an incredible time to be involved in neuromuscular disease. It's changed so much just in the time since I graduated fellowship in 2012. So I also want to thank all the other speakers who are taking time on a Saturday to stare at their computers and reach out to you all, and to everyone for taking time out on a Saturday to listen to us.
Dr. Quinn,
yeah.
Are you going to share your screen?
Is it not sharing?
No, we just see your video.
Hang on one second.
Okay.
We will fix it. Share. Try this.
There you go.
And now let's see if it shares the right screen. Is it sharing the correct screen, or do I need to swap it?
Swap it to presentation mode.
There we go. It's not a virtual talk unless there's a glitch. That's
perfect.
How this has to work. All right, and then I'm going to move my camera because it's opposite where I wanted it to be. So I have some disclosures. I don't, I serve on advisory boards for different companies, apparently only companies that begin with an A, but I don't think any of these are terribly relevant. I do make one mention of a drug that actually won't be moving into commercial production, so I don't consider that a conflict. Also, I wanted to mention Sarepta was kind enough to sponsor today. They're mentioned several times in this talk, but I actually had no idea they were the sponsor when I made the talk. So that is coincidence, except for the fact that obviously they have a vested interest in being involved and have done an incredible amount of work in the neuromuscular space. So what do we want to accomplish today? So first, I think the goal is going to be to kind of look backward, to talk about how things have changed in defining LGMD, how we diagnose LGMD, which has fundamentally changed. And then we'll do the kind of fun part of looking forward. And it's in the past, when talking about therapeutics, you're kind of looking towards a distant horizon, but this isn't really looking that far ahead. We have active therapeutics that are on the market and several that should be coming soon. So we're not looking too far into the future when I say we're looking ahead. So the term LGMD, limb-girdle muscular dystrophy, muscular dystrophies were defined and different patterns were defined back basically in the 1800s. But in 1954, there's this paper in Brain in which they kind of coined the term limb-girdle muscular dystrophy, which just describes a pattern of weakness, meaning that the shoulders and hips tend to be the most involved. Over time, other muscles are obviously involved, but the most significant thing is shoulders and hips, meaning difficulty getting out of a chair, difficulty reaching overhead as kind of like primary symptoms. So that's 1954. As you can see, things have gotten a lot more complicated. We now kind of recognize that there are all of these proteins, and that's what you're seeing in this kind of really busy diagram. There are all these proteins that go into the muscle cell structure and function. And basically, if you have a problem in any of these proteins, the possible result is limb-girdle muscular dystrophy. And so as we've kind of gone protein by protein, and that kind of means gene by gene, we've found lots of different types of limb-girdle muscular dystrophy. Now, obviously, it's really hard to read how many, and this is just to say that there's a lot of them, and you might find your particular limb-girdle muscular dystrophy. Dr. Wickland is going to talk more about the details of different types of limb-girdle muscular dystrophy. So with this many genes, we've undergone a significant sea change in how we approach the diagnosis of limb-girdle muscular dystrophy. So it was not that long ago, I mean, a decade ago, if someone came into clinic and they looked like they had a myopathy, the first thing we'd say is, okay, you need a muscle biopsy. And what were we trying to accomplish with a muscle biopsy? Well, the first thing was we could just basically confirm that the person had a muscle disease, because muscle problems have kind of a distinct look on a biopsy. But then you could also try to find the specific type of myopathy. Now that you had to get kind of lucky, because a lot of muscle biopsies aren't specific. They just look what we call myopathic, and there's no real pattern to it. Now here's an example of, say, centronuclear myopathy, in which that pattern kind of tells you you're in a certain category of disease. But even then, it doesn't tell you the exact gene. And that turns out to matter. So then step two, and this was a little further along, so from the the 90s to the 2000s, we would do single gene testing. So single gene testing is where you're basically trying to piece by piece look at a single gene and figure out, if we're going to talk about what genes are, but if we think of genes as having are made up of DNA, and DNA is a series of letters, we're just looking for basically misspellings or missing sections of DNA, but you have to kind of painstakingly do it piece by piece. That's really expensive. And so what we used to do is try to figure out, okay, let's get in the neighborhood. Let's figure out, what do we think this person has? And we'd send a single gene. And there was no discount for sending, 5 genes. So if it was $1,000 per gene, to send 20 genes was $20,000. So it was really, really expensive. And we just weren't as good at picking the gene as we hoped. So this is the new approach. And this really has changed in the last decade, and really kind of picked up, I would say, in the last 5 to 7 years. So now we use something called next generation sequencing, which it doesn't really matter what that is. It's just you can sequence a whole bunch of genes at one time. And that allows us to basically send gene panels in which we say, okay, we think this person has a muscular dystrophy. Let's send a muscular dystrophy gene panel that covers all of the genes, or most of the genes we think it could possibly be. It's cheap, I know we're talking in the thousands of dollars here, but it's relatively cheap, and there are also several free programs to do these genetic tests. So I think an interesting question is, do you really need a neurologist anymore, right? Can't you just go to CVS and get your gene panel? The problem is that when you send this many genes, you often get a ambiguous result. It's not always a binary yes or no. You have to have someone who kind of understands the diseases to know whether or not you got a logical answer back. And then also, unfortunately for the adults, the most common adult onset muscular dystrophy is actually, for reasons that are not worthy of explanation right at the moment, don't work very well with this technology. So you'd be missing actually the most common adult onset dystrophies. So what's the end result of this approach? I think that the old approach, and again, this was when I was in fellowship a decade ago, we'd say something like this. It looks like you have some kind of muscular dystrophy, let's say a limb-girdle muscular dystrophy. There are lots of overlapping characteristics. It's really expensive to figure out which kind you have, and it's not really going to change how we treat you. So let's just call it limb-girdle muscular dystrophy. And now it's just fundamentally different. The diagnosis is just cheaper and easier. We can send a single gene panel and get back an answer within a couple weeks to a month. And I also think not just that it's easier to do, but it's more significant for the patient, because in the past, if you had a rare genetic disorder, let's just say you lived in a village, you're the only person besides maybe the other family members you have who have the same disorder who has that. And it doesn't necessarily do you that much good to know a bunch of specifics about it. But now, as we see today with everybody calling in to this meeting, you can find someone, no matter how rare your disease is, you can find someone else who has it and share that experience. And I think that that's really, really important for people. The other thing, as we're going to see in just a few moments, is that we have arrived at gene therapy, and that disease-specific treatments are here. And in order to get a disease-specific treatment, you have to know what your disease is. So we really need to make sure that we're providing people with as much information as possible about their disease. So now for the kind of the fun part, all right? So I broke this down for new therapies into two different categories, kind of gene-specific therapies and non-gene-specific therapies. In gene-specific therapies, we'll talk about gene replacement, gene repair, and protein or RNA translation repair. In non-gene specific therapies, we're going to talk about steroids, myostatin inhibitors, and stem cells. So let's just get out of the way that this is going to require a little biology. And don't worry, we're going to work through it together. If biology was not your favorite subject in high school, we're in this together. And also there's no test at the end, so just take in what you can. But we kind of need to describe biology in order to understand exactly what we're up to with these different treatments. So first off, what's a gene? Now we've already kind of mentioned genetics, and I'm sure many of you already know this, but just to make sure we're all kind of starting from the same place. The DNA is the genetic code that is basically the blueprint for everything our cells are and do. And our DNA is made up of a series of kind of chemical letters. And if you think about it, you want to break those letters into little chunks, which we call genes. And those chunks are things that make a specific protein. So those letters kind of tend to be in sequence, and they all blend together to do a specific function. We have 2 copies of most genes. We call versions of genes alleles. And I don't think it matters so much for the rest of the talk, but just because we're talking about it, recessive disease is when impairment of both genes causes disease, and dominant disease is when impairment of one gene copy causes disease. So another thing we kind of have to basically understand is that how do we get from DNA to the end product, which is a protein, right? We go through this middleman, which is RNA. So we transcribe DNA to RNA, and then we make protein from that. And that's going to be important, kind of at what level are we trying to affect the cell's biology. So we're going to go through these 3: gene replacement, gene repair, and protein translation. Technically, kind of the first 2 are often considered true gene therapy, and the third one is kind of not, because you're not really changing the genes, but just for our purposes, we're going to keep them all together. All right, so this is kind of the most basic question that we get. I feel like I get a fair bit. Like, my gene X doesn't work, can't you just fix it? I've heard a lot about gene therapy. What's going on here? Why can't we just replace it? And what we're looking at in the diagram is, we're working at the level of DNA, and rather than kind of monkeying with the core DNA that sits in the cell, couldn't we just add a little bit of DNA? And the answer is yes, sort of, but it's complicated. So how do we approach this? This has taken decades of work, which I'll show in a timeline in a second. But basically, when we're talking about gene delivery, so we want to somehow get a new gene into a cell. It turns out that an effective way to do this, but not the only way to do it, is to put the gene in a virus. Because viruses, their entire job is to get their own genetic material into cells. So we're just harnessing that skill and kind of taking out their genetic material and replacing it with something that we want to put in there. And viruses attack certain cell types, and so we can actually kind of take advantage of that. And over time, we've figured out more and more different types of viruses that get into different cells of interest, like motor neuron cells or muscle cells. And this is kind of a tricky point, but it's important that in this case, the types of viruses we tend to use will create DNA that sits outside the normal cellular DNA. So you're not actually changing a person's DNA when you do this. You're just adding a little bit on the side. And the hope is that with these treatments that you get kind of perpetual production of whatever gene product that you want. We don't actually know how long it lasts, and it requires that the cell that you put it into lasts, but we do have some idea that this can last for many years. So it's taken a really, really long time to get to where we are. And when we look at this, you know, the discovery of this gene, this virus that we tend to use, which is AAV, it was discovered a really long time ago and first used for gene delivery in 1984. But the first FDA approval for a disorder that causes blindness wasn't until 2017. This has taken a ton of time to work through. So why? You know, it seems like, all right, 1965, we had it pretty well sorted. In the 80s, you got your first gene therapy. It's 2020. What's the problem? Well, the first issue with any therapy is you don't want to hurt people. It's part of our oath that we're not supposed to do damage while we're trying to do something good. And this was kind of, you know, brought to the fore right at the advent of gene therapy with the death of Jesse Gelsinger, who had a liver disease and underwent gene therapy and died. And the problem with gene therapy is you can't take it back. If it's not going well, you can try to manage the medical problems, but you put that in, you put the gene in there, you can't take it out. So we have to be pretty cautious. And while there were huge changes to how we approach gene therapy, and we use different viruses that are less provocative to the immune system now, we still have this problem. You know, there was just recently, you see this is from June 29th, this announcement that there was a trial in a muscle disease in which 2 children died from liver problems caused by a gene therapy. So, you know, the problem of unintended consequences and not being able to take back the therapy once we give it, you know, causes us to be really, really cautious and really focus on safety. Another issue is gene size. So the little spiky thing that you see in the center of the screen, that is, you know, a cartoon of AAV, which is just a type of virus, and it's the most commonly used virus in gene therapy. Well, that virus is a specific size, and you can only put so much in it. And what I've mapped out, so the little green and yellow thing is a piece of genetic material. And it turns out that it can only be what we call base pairs, are just the letters, right? So this can only be 5,000 base pairs long. Well, unfortunately, a lot of the proteins that we're trying to replace are huge. Dystrophin, which causes Duchenne muscular dystrophy, is massive. It's 2.3 million base pairs. Now, fortunately, we can get down to a kind of the natural basic version of dystrophin, which is much smaller, so 11,000 base pairs, but still that's twice as large as what you can fit in AAV. We're going to talk about how do we work around that. Another issue is transmission efficiency. So, you know, when you get a cold and a virus goes into your respiratory system, it doesn't infect every single cell. And in that case, that's good. But for our purposes, obviously, if we want to try to have a positive impact on muscle cells, we want to infect as many of them as possible. And we don't. So we're constantly trying to figure out how to increase this efficiency. It's also super expensive to do these types of trials because it's very expensive to make virus. So we've recently been looking at a kind of starter gene therapy trial. So 10 patients, just the trial, not all the science and the lead up, is $10 to $12 million just to do this little bitty trial. And then, of course, on the back end, that means that therapies are very, very expensive. So this therapy that we're talking about here, $2.1 million for a single treatment. Again, the hope is that that's a single treatment, and, you know, rather than having the cost spread out, it's just all up front, but still it's ridiculously expensive. All right, so how do we get around this size issue? Again, just going back for a second, so we have this problem where a virus is a limited size, and a lot of the proteins that we want to work with are too big. We kind of have 2 strategies for this. Again, this is kind of a busy diagram, but I just focus on the second and third line. So one option is that we just use a smaller version of a gene. What is the kind of bare essential version of a gene? We don't cut off the ends as is kind of diagrammed here. We try to select out the portions of a gene that we really, really need and package that up in a virus. Another kind of cool strategy is that we could break a gene like dysferlin into 2 pieces and then put that into 2 separate viruses and then infect someone with both. And we're going to talk about both of these. So busy science slide here. Don't worry. Let's focus first on the left-hand side. So this micro strategy has been used in Duchenne. I'm going to focus, I know Duchenne technically is not in the limb-girdle category here, but it's the most common muscular dystrophy, so it's often a good starter point, and it has a lot of implications for our approaches to limb-girdle muscular dystrophies. So in this case, the top image, the top cartoon on the left is normal dystrophin, which I said is really, really huge, the biggest protein that we have. And then kind of creatively, it's been trimmed down, and there's lots of different approaches to trimming it down. So there's lots of different, what we call microdystrophin, in which people try to figure out, okay, what are the bare essentials we need in order to get a protein that works? And this was tried by Dr. Mandel, who's been at the forefront of gene therapy for a really, really long time, in 4 patients here. And basically the top line is that showing that they don't have dystrophin. And the bottom line, the red, is cells that are expressing dystrophin. And so it seems like this works. We can kind of use the small version. And there's actually a phase 3 trial that just was announced pretty recently for Pfizer for a different microdystrophin. So a different kind of version, miniaturized version of the protein. So that's really, really exciting. A phase 3 trial is the last phase before approval. It doesn't always mean approval, but it's as close as you can get without having an approved drug. All right, so this one's a little weirder, and but still really cool. It's not as far along. So dysferlin, I'm sure there's some LGMD2B people on there, or LGMDR2 if we're using the new nomenclature. Dysferlin is just a little too big. It's like 6,500 base pairs, right? So the max is 5,000. So the idea here is, what if we could just split the protein in 2 and put it in 2 viral vectors and infect someone with both? I know it sounds odd to say infect someone in a positive way, but in this case, it is a positive thing. We want these viruses to get into the cells. And here what you're seeing is that if we treat a mouse, again, this is at the mouse level, we're not to people yet, so it's not as far along as microdystrophin. You see that the untreated is on the bottom, and these cells look really, really unhealthy. And there's lots of scar tissue, and they don't express dysferlin. But then if we treat them with these 2 viral vectors, we see that the cells look healthier, there's less scarring, and they're expressing dysferlin. Sounds easy, except look, there are some issues here. We mentioned efficiency. So now you imagine that it's not just that you have to get 1 virus to go into a cell, you have to get 2 viruses to go into the same cell, and they have to express properly, and they have to meet up. It's kind of a miracle that it works, but it seems like it might be a good strategy. So this is just a slide to show you. So on the left are all of the different, and this is out of date because this is from a presentation I gave a couple of years ago. All of the different gene therapies in lots of calpain, dystroglycan, sarcoglycan, dysferlin, there's lots of different gene therapy programs in lots of different LGMD. And again, Sarepta, they did a sponsor today. I didn't know that when I made this slide, but they're just really active in this space. And they have lots of different therapies for sarcoglycan, dysferlin, anoctamin, calpain. And what you see is these bars indicate how far along are they. And when things are in the discovery phase, that means, chances of working out aren't that great, quite frankly. We have a lot of failures before we have success, but a lot of these bars are up to, clinical trials, and even some of them into deeper clinical trials. So a lot of these are really close to this end, which is commercial, meaning that they're actually available for people to get, which is really exciting. Okay, so the next thing we're going to talk about is gene repair. And I think this is probably for the more, you know, the people who kind of spend some more time looking into genetics. People get really excited about CRISPR. And I've put a horrible pun in here. I apologize. It's like mid-talk, so you have to have a horrible pun. We've got some ripped genes. We add a little apple crisp, and, you know, presto changeo, we have fixed genes. That's not exactly how it works. What we're trying to do here is actually, rather than focus on, you know, adding in a gene that's parallel to the genes that are there, we're actually repairing the core DNA. We're getting into the nucleus, and we're changing the DNA. How do we do that? Well, we use this guide RNA. So again, RNA is this thing that DNA gets translated into, but it also matches the DNA. So we use a guide RNA to kind of match up with a sequence of letters in DNA that we care about. And that brings along this thing called Cas9, which acts as a pair of scissors that actually cuts the DNA at a very specific target. And that allows you to basically take out things you don't want, so you can snip it and let it kind of repair itself, or you can even add in things you do want, so you could replace something. It's cheap, actually, as far as, you know, these sorts of things go. It's quick. It's pretty easy to work with. I'm not a scientist. I don't have a bench, but my understanding is that, you know, this is something that's pretty easy to work with, and it's pretty precise. Where it cuts is pretty precise. So where are we with this? Because, you know, we hear so much about it. Well, the fact is that we're not to people yet. This is a paper just showing Duchenne CRISPR in mice, and so it's really exciting. It looks like it might work, but there are currently no human trials in neuromuscular disease. Why are we, you know, if this is so promising, why is it not further along? Well, one thing is there are safety concerns. So this is a molecular cutting machine, and you only want to cut where it's supposed to cut. And while it's pretty precise, we want it to be really, really precise if we use it in a person. The other thing is that we're changing someone's actual genome, the DNA, the kind of core DNA that makes us what we are. We're changing that. We're also changing the transmission DNA, what DNA goes to children. That's a really concerning approach. It may be promising. It's not worth pursuing, but I think everyone has kind of appropriately proceeded with caution. All right, so rather than change the DNA, in this case, we're going to look at how could we manipulate this middleman to get what we want, to get effective protein. So in this case, we use something called antisense therapy, which, again, horrible name, sorry, I didn't invent it. But basically the idea is that when you go from DNA, which has a genetic code, and that gets transcripted into RNA, again, this middleman code. And the kind of fun thing about RNA is that you can make this kind of flipped copy that can stick to that RNA. And that allows you to manipulate it. You can patch it. You can reduce its expression, actually increase the protein production or improve a protein, all through this technique. And it's a little confusing to try to get into too much detail, but it's also important to talk about because these therapies are already out and available. This is kind of the technology that's the furthest along. So many people probably have heard of eteplirsen, which is an exon skipping approach. Well, what is exon skipping? Basically, in Duchenne muscular dystrophy, the problem is that you have a mutation that causes the RNA to not fit together properly. So you get this truncated protein. Basically, you have a problem in the RNA. The two remaining pieces that are on either side of that problem don't fit together, and you don't get what you want, which is this dystrophin protein. What this skipping does is allow you to kind of stick to the RNA and cause the piece that makes the RNA not fit together get patched over. So now the RNA does fit together. And when you can put these two pieces together, there's a milder form of Duchenne in which you don't have normal protein, but you don't have this really truncated protein, and that is a much milder form of disease. So you've created this little patch that helps stick things back together, and you get a shortened but functional protein. So what are the issues with this? Well, first of all, let's just say that there are already approved. As you see, we've got the purple bars that go all the way to commercial here. So that means that exon 51 and exon 53 skipping is already approved. But the reason why there's an exon 51 and 53 therapy is because these are mutation specific. Now, not all antisense therapy is actually mutation specific, but in this case it is, which means it's a painstaking process to go through each form of skipping. And so each individual therapy covers kind of a small batch of people. And it's not very efficient in terms of the protein translation, and that may limit its clinical effect. And also you have to give kind of persistent treatment. It's very expensive. All of these therapies are expensive. It requires persistent treatment because you haven't added a gene or changed the genome. You're kind of constantly having to put this patch on, and that only lasts so long. I just want to add this in because we've talked so much about dystrophin, and that's not technically a limb-girdle muscular dystrophy, and this is using the same idea in sarcoglycan. So basically there are other kind of non-gene therapy approaches that are specific, such as like enzyme replacement for Pompe disease. There's way too many to talk about here, but this just emphasizes that knowing your genetics is really, really important because you don't know what to look for if you don't know what your genetic issue is. All right, so I'm going to try to move somewhat quickly. These are fast, like one slide each, okay? So steroids have been used for forever in Duchenne muscular dystrophy. This paper that's on the top left here that is, says 2016. It's really data from the 1990s. We've been using steroids for Duchenne muscular dystrophy, and it shows that it prolongs walking in Duchenne. There's reason to believe that it might have similar impacts in certain LGMD. So in FKRP, there was a trial of a steroid trying to look at whether or not it could have the same effect. Unfortunately, there was under-enrollment, and that trial was actually closed, but still, the logic holds. And then this is another one of these scary science slides, but myostatin inhibitors have been popular in muscle disease for quite a while. What is a myostatin inhibitor? Well, we think of myo as muscle and statin is stop. This is basically a double negative. So we're stopping the thing that stops muscle growth. And the cool thing about that is that could be applicable to lots of different muscle diseases. But the problem has been it's tricky to really use. And with systemic delivery, meaning that you say give someone this medicine by IV, it's hard to change the impact on muscle enough, so to make the muscle grow enough to be stronger. But then there's recently a trial. This is the one that we were involved in, where you directly inject actually in this case folstatin inhibitor, but similar idea, directly into the muscle, where you actually do get much more muscle growth and improvement in strength, but it's hard to impact function when you're injecting one muscle at a time. So unfortunately, the myostatin inhibitors, there's a lot of promise and a lot of excitement, but most of these programs have failed. I get asked a lot about stem cells. I work with patients with ALS and with muscular dystrophy, and I was thinking for this talk that we don't talk about this much with muscular dystrophy, and I really honestly don't know why. I had to kind of do my own research because stem cells just aren't that much of a thing in muscular dystrophy. And as early as 1995, you see this myoblast transfer in treatment of Duchenne muscular dystrophy. So myoblasts are these early cells that will go on to become muscle cells. And the idea is that if you could give either satellite cells that become myoblasts or myoblasts that go on to become muscle cells, maybe you could get effective muscle replacement. But I think it's just been tricky in practice. It's hard to get the cells, enough cells where you want them to go, enough places to really have an impact. And some people might be aware of the fact that there are stem cell trials in ALS, but in other neurodegenerative diseases, you're really not doing what you're trying to do here. And here you're trying to replace a cell type with the same cell type. In ALS, for example, stem cells are really kind of a drug delivery model. They're not trying to replace neurons. They're trying to make a healthy environment for neurons to live in. So it's really a different thing. So what are the barriers to progress? Well, the first thing is that these are rare diseases, but I will argue that the only way we're going to kind of find enough people is to make sure that everybody who has muscular dystrophy knows exactly what type they have. These studies are really expensive to perform. We know this from personal experience here at Penn. It costs a lot of money to try to do these studies. We don't have a lot of outcome measures, especially, I should say this is adult biased. And that's important for trying to figure out kind of whether a therapy really works. And there's, especially in adults, there's a kind of lack of natural history data. These muscle diseases tend to change slowly over time. So it's hard to tell if you're having an impact if a trial is only 6 months long. So I think looking back at the last 10 years, our ability to kind of find a specific LGMD has grown tremendously. And I think, I put over the next 10 years for kind of parallel structure, but I really think over the next 5 years, we're going to see the development and, successful trials of multiple specific disease treatments for multiple limb-girdle muscular dystrophies. Again, I want to thank the MDA for the opportunity to speak with you all, and I'm happy to, I think I left enough time to answer some questions here.
Yes, you did, Dr. Quinn. Thank you very much. And we have had the questions come in too. So thank you for utilizing the webinar chat feature. I just wanted to remind everyone, make sure you click to respond to all panelists and attendees. That way all the attendees can see your question. Okay. I'm just going to ask you a few questions that we've got. Is the original testing now done only by blood work, or is there a biopsy that's done?
So it depends. I feel like every doctor answer starts with it depends, but it depends. A lot of the gene panels we can actually do using saliva, but it's usually saliva or blood. Sometimes, like for mitochondrial disorders, we end up using actual muscle tissue, but the vast majority of the time, I walk into clinic, I have a little kit in a cardboard box, I hand it to the patient, they spit into a tube, and I personally FedEx it.
Okay. Here's a question around LGMD 2A/R1. Is there a possibility that any of my kids will have it? We all have had some muscle weakness concerns with my son, and the doctors are currently watching closely.
Yeah, so usually with recessive diseases, the chances of passing that on are quite low, right? Because dominant diseases, the concern is that each generation has kind of a 50-50 chance. But usually recessive, which is the majority of LGMD, right? Most LGMD is recessive. It's kind of spotty in a family because you have to get two people who have the same genetic carrier status to match up and cause disease.
Okay. Is Dr. Quinn saying that gene replacement means that my diagnosis can be reversed even though I was diagnosed at age 10?
That is a fantastic question, and I'm sorry. I'm actually sorry I didn't kind of explicitly state that. So I don't think that we're looking at disease reversal. I think, we don't know exactly what the limit is, because a lot of these therapies are tried in children, in which you think that the amount of, when you think of a dystrophy, what defines a dystrophy? The muscle is injured and gets turned into kind of scar over time. As we saw on one of the slides, there was this scar tissue. I don't think once a cell has gone to scar tissue, is it going to come back to muscle? No. Is it possible that we do get enough improvement in muscle health to actually show improvement in strength? I do think that's possible, but I think the idea of kind of normalizing people who've had muscular dystrophy for a long time is probably not what we're aiming for.
Okay. Here's a very interesting question. If a teenager wanted you to explain how the genetic process of limb-girdle works, how would you explain that to an 11 to 14-year-old in the terms that they could understand?
Yeah, so first of all, that's a fantastic question. I have a 7-year-old. I will practice when I go home and see. I do make her listen to me an awful lot about these topics, so we'll see how successful I am. But, I think what we talk about is going back to that picture of all of these proteins that keep the cell structure. Muscles undergo an enormous amount of stress. Their cells on the move, right? And when we affect these proteins that stabilize the cell or affect even cell repair, when they're stressed over time, they break apart. And our body does the very best it can. It doesn't just take that line down. It tries to generate new cells and keep our strength up, but there is a point at which you can't keep up with that process. And there's just too much muscle cell injury, and that's when you develop weakness. I hope that that's what you're asking, but that would be my answer.
Okay, perfect. We've had a couple of people ask, how does, and I'm sorry if I'm not going to pronounce this correctly, how does levocarnitine help us?
I actually don't know. Okay. That's a great question. I don't know.
That's fine.
Carnitine has been studied in different diseases, but I'm not sure how when you say it does help, I'm not sure that it does help, but if it does, I'm not sure how.
Okay, perfect. Let's see. This person has a question related to her diagnosis. It's limb-girdle Layman AC type 1, autosomal dominant. Is there anything on the horizon for that?
I haven't looked, so I actually have a fair number of Layman AC patients. I haven't looked at it in a little bit. Like what I would encourage people to do, because the fact is that I take care of all of the letter, that alphabet soup that you saw on the slide. I am responsible for all of those genes, and I do my very best to keep up with what's the latest, greatest. But I actually really rely on my patients to tell me, like, hey, I heard this cool thing about protein X, their protein of interest. And so I really would encourage people to use clinicaltrials.gov or the MDA equivalent, and bring it to us, because you are the expert on you. You are the one who are going to know, the moment someone tweets out that there's a promising therapy in Layman AC. So I don't know of one right now, but my hope is that actually you will be the one to know, and you're going to come back and circle back to us.
Okay. Regarding Sverlin, which one of the treatments are the promising in your opinion?
Well, I think the one that's been kind of brought, is getting close, is this dual vector idea, right? Where Sverlin is the one that's just a little bit bigger than AAV, and we're splitting it into two. And I'm hopeful that that's going to work. Now that it's a tricky thing to do, but that's I think the focus at the moment.
Okay. Why is treatment with deflazacort for limb-girdle considered closed or non-effective?
So it's just that the trial was closed. So thank you. Oh. That's actually really helpful. So what happened was is that I think—
Perfect. Thank you for—
yeah, the company had a kind of very ambitious enrollment goal for FKRP, and they just weren't able, and let's be honest, COVID didn't help. They weren't able to enroll as many people as they wanted. So they didn't close it because it didn't work. They closed it because they realized they weren't going to be able to answer the question, and they don't want everyone to go through all this if at the end of the day they can't answer the question they sought to answer.
Okay. Do you recommend any supplements for limb-girdle patients like CoQ10, carnitine, others? This person is dependent on the type, or is it dependent? Sorry, is it dependent on the type of limb-girdle? This person has type 2L.
Yeah, I don't. I know that varies. I've seen very different things. I don't think there's enough evidence to recommend specific supplements. I don't discourage people. I mean, I recognize a lot of times we're not providing a fantastic alternative. So I don't claim that we know everything. And I do recognize that there's some science to support some supplements, carnitine, things like that. But I don't think there's enough science to actually endorse it.
Okay. Do you know if there's anything being done for type 1B? There is not much mentioned in materials around this diagnosis.
I'm sorry, I just didn't hear the type.
1B as in boy. Is there anything being done for type 1B?
I'm not sure about 1B, to be honest. And I'm sorry for those of you who have these specialized types. It's hard for me to keep. I don't want to say there isn't something, because it's very possible that some scientist out there is screaming that they've dedicated their lives to 1B, but I don't know of anything that's close.
Okay. So this person wanted to just kind of clarify, and maybe you could speak to this a little bit more. Treatments are currently to stop further weakness, but not to repair the muscle. Is that accurate?
I think it's probably a little bit between the first question related to this and this question. It is possible that we get improvement in strength, right? I just think the amount of improvement may be limited by the amount of scar tissue there that's already there.
Okay. Let me check our time. We have 1 minute. We'll close out your session. This actually is a very, very good question. What is the best approach for the future if you have been undiagnosed for many years and have had many different tests done and come back negative? What would you recommend that person do? I feel like a lot of people might be in that boat.
Yeah, so I think it kind of depends on what's been done. Okay. So, we have lots of patients who show up to our clinic who are in that boat. Our goal at this point is that first of all, we have to kind of convince ourselves that it's a muscular dystrophy, because sometimes the issue is that we're just barking up the wrong tree, right? So the first thing is to make sure that we're convinced that there's a muscular dystrophy. We usually send one gene panel, and if that doesn't produce the result that we want, we sequence the entire exome. And it's very common to do that. Insurance covers it usually. That probably varies from state to state. And then if that doesn't yield an answer, that's sometimes when we go to a muscle biopsy and do some specific genetic testing on a muscle biopsy.
Okay. Well, thank you very much, Dr. Quinn. We appreciate your time and all the help and advice that you've given everybody on our panel and all of our attendees. So thank you very much. Have a great rest of your day.
Thank you. I really appreciate the time.
And everyone, I wanted to make sure that you guys were also aware that coming up, we will begin at 12:10. Eastern time, and we will begin with a genetics discussion and Dr. Matthew Wicklund. So stay tuned. Thank you.